Okay, great. All right, so again, the title today is Next Generation Pgta with NGS or Next Generation Sequencing. Here are my disclosures. They're also on the website, so I'm not going to belabor those at the moment. I think it's important to start this, anytime we talk about Pgta, with this sort of brief foray into the past. There's a lot of names that this technology has been called. Pre-implantation genetic screening, or PGS, is the one that you're probably most familiar with. There was the use of comprehensive chromosome screening, which sort of took hold a few years ago when some people wanted to sort of separate themselves from doing PGS, perhaps. As a matter of fact, I was just reviewing abstracts for ASRM today, and someone's still using CCS. There's other names. I have a few here: aneuploidy screening, whole chromosome screening, etc. In 2016, the World Health Organization harmonized those terms to the following. And we need to stick with this, because the more we use different terminology, the harder it is for everyone to keep up with what's going on. So we're talking about pre-implantation genetic testing for aneuploidy or Pgta, right? That's what it should be. So it should be Pgta always. And I also like to just sort of review a little bit about the Pgta, the evolution of Pgta, because I think it's important to understand where we came from and where we are. Certainly in the early 1990s into the mid 1990s, we were using multicolor fish to look at a few chromosomes. This has been termed sort of first generation Pgta. I think Santi Monet is the one that sort of started calling newer methods Pgta 2.0. Jacques Cohen did point out to me that there's sort of a hybrid in between here. There were a couple of groups that were using fish but doing all 24 chromosomes. But for the sake of simplicity, I like to think of it as multicolor fish. Even for 24 chromosomes, it was really difficult. That's first generation Pgta. And then everything since then, which includes metaphase CGH, array CGH, qPCR, SNP arrays on here, and anything to do with next generation sequencing NGS, I think is sort of second generation Pgta, because we can very easily and concisely look at all 24 chromosomes at one time. And that's the hallmark for me of Pgta 2.0. So to go from there, let's talk a little bit about next generation sequencing. And so this is Jim Watson, he of Watson and Crick, holding a hard drive in May of 2007. That hard drive has Dr. Watson's entire his entire genome is embedded on that hard drive. So that's the basis from the top of chromosome 1 all the way through the bottom of chromosome Y, and every base in between. It's about 3 billion bases. Jim Watson was one of the first 2 people in the world that had their entire genome sequenced. J. Craig Ventnor was the other one. He was the guy that owned the company that did most of that sequencing. The reason I'm telling you this is that back then, in 2007, that project took 15 years to complete and cost about $3 billion to get 2 genomes. 15 years, $3 billion. The reason we're talking about next-gen sequencing today in IVF and in PGT is this exact reason. Sequencing companies today can do that same exact trick, sequence your entire genome for about $1,000, and it takes less than a week to get done. So like most things with any technology, we've gone from 15 years and $3 billion to 7 or 8 days and $1,000. And it's just going to keep getting cheaper. And so I think it's not only going to be PGT that you see sequencing in, certainly if you look at the COVID things that are going on, it started with PCR tests, then serology tests. Now we've moved on into next generation sequencing tests to look at where where the virus came from, et cetera. So next gen sequencing is creeping into every bit of medicine as we see it. And so I think to talk about next gen sequencing, we really need to start at the most difficult part of all this, which is the biopsy, right? I can't underplay how important it is to do good biopsy and to handle the sample really, really carefully and well until it gets to the Pgta laboratory, and that's going to create the best results. And that's what my co-speakers are going to talk about today, is things that go on in the lab that might affect the outcomes. But before we do that, we're going to talk about how we actually get a result. So you do a biopsy, we get a few cells. In those few cells, there's a little bit of DNA. There's not enough DNA in there to do anything really fantastic with. There's certainly not enough DNA there to do next generation sequencing and get a real result. So we take the embryonic DNA, and we do whole genome amplification, or WGA. And there's a million ways to do it. I'm not gonna talk about that, but let's just presume you do some sort of amplification, WGA or a targeted PCR. And so what happens then is you end up with then lots of DNA. And now you want to get that DNA ready to go on the sequencer, right? So you take your unknown DNA, you fragment it down to a certain size, and then you need to put what's called adapters on there. And those adapters are nothing more fantastic than little pieces of DNA that each sequencing system uses to move the DNA in and around the sequencer, because DNA doesn't just go into a tube and get sequenced. It goes onto a hardware, a hard platform. So adapters are simply pieces of DNA that are used to move DNA around. So the amplified embryonic DNA is fragmented into smaller pieces, and then adapters are put on, and you have then what's called sequencing ready library. So you then load all that DNA onto the sequencer, and the sequencer does what? It just sequences. It sequences every region of the genome multiple times, over and over and over again, right? So the typical sequencer that you see in a Pgta lab globally today, you average about 25 million reads, meaning that there's 25 million fragments of DNA that are sequenced, right? And so we have to do then something with these literally millions and millions and millions of short sequences. So the first thing we have to do is align them. We have to figure out, where does it belong? So there is, in the end, there is a human genome. It's called the HG19 genome. It is the one genome for all humans, even though we don't all match up to it exactly. But there is one reference that we all agree to as the reference. And so you take each piece of DNA, you notice the orange and blue adapters on the ends. The sequencer forgets those and just reads the gray bar down the middle, which is the unknown sequence, and it aligns, right? It says, where does this piece of DNA belong in the genome? Does it belong on chromosome 1 or on chromosome 6? And it assigns it then to which chromosome it belongs on, but not only just which chromosome, but where on the chromosome it belongs, which means it might be putting, the first piece might be on the P arm, the very top arm of chromosome 1. The next piece might be right near the centromere on chromosome 8 or wherever else. And so that's what's called alignment. Now, if we did what I just described, for each embryo, it would, number one, take forever, and sequencing will be quite expensive, because obviously there's a raw cost to do sequencing. So some really clever people came up with this other idea called barcodes, right? It's not unlike the barcode that you use when you get on an airplane that says this is Gary Harton's seat. He's sitting here and he's meant to be on this airplane going to wherever. Or if you go to the grocery store and you buy a loaf of bread, the scanner knows exactly how much that loaf of bread costs. So we use DNA barcodes, which allows us then to combine multiple embryos into one sequencing run, and multiple embryos from multiple patients into one sequencing run. There then you're taking advantage of this 25 million reads. You can spread those across hundreds of samples, right? Depending on how big your sequencer is and what your throughput needs are. There's 8 sequences all the way up to hundreds of samples, maybe even 1,000 samples on a much larger sequencer. And so then you go adapters on the outside, because we still need to move the DNA around, barcodes next, then the unknown piece of DNA in the middle. So then we do the same thing. We make libraries, put the barcodes on, sequence. And then when we do the alignment, not only are we figuring out where in the genome does this piece of DNA belong, is it chromosome 1, chromosome 2, chromosome 3, but also where does this DNA come from? Is this Mrs. Jones's embryo 1, or Mrs. Jones's embryo 2, or maybe it's Mrs. Smith's embryo 4, right? Because we can combine multiple embryos from multiple patients into one sequencing run, and therefore really harness the power of next generation sequencing. So once you've aligned all of these pieces of DNA, you're going to align them and you're going to place them into a bin. And a bin is simply where we take the genome and break it down into usually approximately 1 million base pieces of DNA. And then you simply count across those bins. So you see here on the very top, Those are the bins, and you can see there are variants from different places in the genome, all along the chromosome. And then each of these bins gets consolidated down into a dot that you're typically used to seeing when you look at one of the profiles that a lab might show you, or that I might show you when I'm doing a talk. So you can imagine there's a lot of bioinformatics that goes into whole genome amplified DNA, sequenced, aligned, cut down into bins. Those bins are counted, and that count is represented by a dot somewhere along the chromosome. And that's essentially how next generation sequencing works. So once you've got all those dots, then the system compares those dots to something, right? So this is the way Illumina does it. Thermo Fisher does it very, very similarly. What they've done is they've taken some number of normal people, they've taken some single cells from those normal people, sent them through the whole process, whole genome amplification, library prep, sequencing, alignment, counting, binning, all that, and come up with a representative number of how many counts along each chromosome one should get for any one chromosome. And you can see here, chromosome 1, chromosome 2, chromosome 3, they all have different counts based on how many genes they have, how gene-rich they are. And then the unknown is compared to the known, blue to orange here. And you can clearly see down here on chromosome 22 that there's way too many counts on chromosome 22, and so the system can then call that a trisomy, right? But again, remember that in sequencing, we're not only counting how many are there in each chromosome, but we're also counting where they are in the chromosome. So next-gen sequencing allows you really to get, pretty good resolution. Some platforms can get down into the 2 megabase, 3 megabase resolution routinely, whereas with array CGH, we're more likely at around 10 megabase resolution. So this makes chromosome rearrangements a little more interesting. You can see more segmental chromosomes. And because of the power of the fact that you're actually counting chromosomes, this has led to the ability to also see mosaicism, which we're not really going to talk about a ton today, but we're all aware of mosaicism, and that it's a particular thing that NGS allows you to see. So you take those chromosome counts. In most softwares, you can drill down to the chromosome level. So here you can really see where each of those pieces of DNA, where those bins are assigned. You can see that they don't actually come out in these perfect nice lines that we like to show you. The real scientists in the Pgta lab, this is where they go when they want to really look at a chromosome and see how it's impacted. But you can see, because of the resolution, the system then allows you to look at, for this is a translocation, a gain or a loss on the arm of the chromosome, and you can see exactly where that gain or loss are delineated, either for a de novo segmental or in a translocation case. And then again, this is what that looks like when we show it to you. Of course, this is a good looking one, but if you look really carefully, you can see there are hundreds of dots along every chromosome, and every one of those dots is a representation of the counts from the bins from all that sequencing that we did. And then you can simply look across and see here very clearly there's a loss, a segmental loss on chromosome 1 and a segmental gain on chromosome 11, simply because this is from a translocation case. And so hopefully in about 10 to 11 minutes or less, I didn't time myself, that's how next generation sequencing works. And I thank you very much for the time, and I thank you very much, Peter and Jacques and the rest of the i3 team for inviting me along today.
Thank you, Gary. That was